My name is Shannon McAmis, and you've probably seen my name on all the emails from the Eventbrite, and that's because I'm the student coordinator for the Florida First Detector program, and I'm super excited to be able to put on the first Florida First Detector workshop for 2021, so exciting new year. And I'm really excited to be putting on a workshop for Duval and Nassau counties, and I'm going to take a couple minutes to introduce the speakers and some of the group leaders that we have today. But before I do that, Larry, was there anything that you wanted to add?
No, I just think it's exciting that we're the first detector session and for 2021, and it's good to see everybody. And maybe one day next year we can get together in person.
Yes, I think that would be great. I haven't had a in-person workshop since I started coordinating the program, so I'm excited to do that one day. And I believe most of you know Larry Figart. He's the extension agent in Duval County, and I think he's wearing a bunch of different hats right now, but so he'll be talking about DDIS today. And the next speaker I wanted to introduce is Dr. Amanda Hodges, who is a faculty member in the entomology department and is also the program director for the Florida First Detector program. Dr. Hodges, was there anything you wanted to say?
It's great to be here with everyone. Thank you so much for joining us today. And we're excited to talk to you about invasive pests, and really looking forward to today and interacting with you.
And then the first speaker you'll hear from today is Dr. Deah Lieurance, who has been the coordinator for the IFAS invasive, or I think I said that wrong. I think it's the non-native plant assessment survey. And she's been coordinating that for 8 years. And so she's going to be talking about how they evaluate exotic plants for invasiveness. The next speaker is Brad Danner, who's the state coordinator for the Cooperative Agricultural Pest Survey, so CAPS for short, if you ever see that abbreviation. And he's going to be talking about the Asian longhorned beetle. And throughout the workshop today, we're going to be having a couple different breakout groups where we'll break into smaller groups. And we have a bunch of great group leaders, and I briefly wanted to mention them. And the first one is Dr. Gideon Alake, who is a postdoc in the entomology department here at UF. And then we also have two Sarahs. So the first one's Sarah Burkmeyer, and she is a graduate student in the Doctor of Plant Medicine program. And then we have Sarah Taffel, who is a student in entomology at UF as well. And last but not least is Trevor Forsberg, who recently graduated from UF and has been helping us out a lot in the program. And so that's most everybody will be talking about today. And before we break out into our first breakout group, a couple technical things to go over. The first one is we'll have times for questions after each presenter. So you can unmute during those question sections and ask your question. And alternatively, you can type up your question and put it in the chat. And either somebody will respond in the chat or we'll ask the speaker at when we have time for questions at the end. And I think someone just also mentioned in the chat bar, mute. So we're going to be recording the speakers. So if everybody could please stay muted during their presentation, that would ensure sound quality. And but feel free to unmute at the end when there's time for questions, and also when you're in the breakout groups. And we don't record the breakout groups, so don't worry about that. And with that, I think we're ready to go into our first breakout icebreaker session. Okay, so I think with that, we can go ahead and transition into our first talk, which will be Dr. Lieurance.
All right, thank you for the invitation. I'm really excited to talk about, the UF IFAS assessment. I've been told by folks at IFAS Communications, we have one of the best programs on campus that nobody's heard of. So any opportunity is welcome to talk about what I do. I am an extension scientist within the agronomy department at University of Florida, and I've been working in this program for about a little over 8 years now. Before I get started into what we do at the IFAS assessment, I just want to give you a brief background in the process of invasion. So it's a stepwise process. There's 5 steps, and there are barriers at each one of those steps. So for a species to become an invasive species, first it has to be transported from one place to another. It has to then be introduced. Then it has to establish. So here you think about things like climate matching. You know, if it's a species that can only live in the tropics and you put it in, temperate parts of the world, it's not going to maybe do as well. Next, you have to spread. So think of this bird eating one of the fruits, flying off, digesting and depositing the seeds. And then finally, you have to have impacts. And those impacts, those can be to the environment. So for example, changes to, fire cycles or reductions of biodiversity. The impacts can be economic. So the citrus industry right now is currently being ravaged by, a disease that's vectored by an invasive insect, or it can be to human health. And there's examples of plants that you might brush up against and get a pretty nasty burn on your skin. But the other important thing to note here is that not all introductions result in invasion. So each one of these steps, there might be different factors that would halt that invasion. The next graphic I like to show is what we call, this is a pretty classic example, the invasion curve. So here you'll see at this point, the entry of a non-native species. And as you move through time, the area invaded will increase and so will those management costs. The curve is broken up into four sections. You have your prevention, eradication, containment, and control and management. And when you get up in this control and management section of the curve, the odds of eradicating that species are very, very low, and it's very expensive, and you have a lot of environmental harm. So we'd like to stay down here in this prevention eradication part of the curve. So if we can identify something either before it comes to the state or as early as possible, we can save money, protect the environment, and reduce those general impacts of invasions. And that's where the IFAS assessment comes in. We specifically work in plants, and we have 3 tools that we use to try to stay at that lower end of the curve. So first we have the predictive tool, which evaluates the risk of a species becoming invasive. And ideally we do that before it enters the state. The second tool is the the status assessment. And this evaluates species that are already here. So think of this as a diagnosis versus a prediction. And then the third tool is the infraspecific taxon protocol, which we shorten it to the ITP. And this evaluates the invasion risk of cultivars, specifically cultivars where we know the parent species are problematic invaders. So just I'll briefly go through each of these tools. The predictive tool is a risk assessment protocol that was modified for Florida. It's based on sort of the gold standard Australian weed risk assessment. It's been tested over multiple geographies, different scales, and it's for as simple as it is, it's very accurate. We've altered it to a few questions to sort of reflect Florida's climate, precipitation, and soil types. And again, we want to look at species that are new to the state. In most cases, we know that they cause problems elsewhere. There is this example of proposed for a new use. So this is written into Florida, a Florida permitting rule, so called the non-native planting rule. So anything more than 2 contiguous acres requires a permit from the state for non-native species, and part of that permitting process requires one of these risk assessments. And then to this day, we have just over 200 species, maybe about 215 at this point, of these weed risk assessments that are available on our website. It's a series of 49 questions based on history, biogeography, life history, ecology. The way you answer those questions, it might give you, whether you say yes or no, a -1 to 1 score. But some questions are weighted heavier than others. But you take that cumulative score, and anything under a 1 is low risk, 1 to 6 is evaluate further moderate risk, and over 6 is a high risk for invasion. The next tool is the status assessment, and please note that we've divided the state into 3 zones. So for example, if you're a plant living in South Florida, you're experiencing a very different climate than those living in North Florida. So you can have a different conclusion for each of these zones based on the evaluation. So we do use this for species that are already in Florida, and we look at the ecological impacts, the potential for expanded distribution. So is this thing spreading? How hard is it to manage? Is it expensive? Does it require retreatment? Are the populations hard to get to? And then in some cases, we do consider the economic value of species to Florida's economy. And we have about 550 species in our database using this tool. We regularly reevaluate those species. So we want to incorporate data from the field from land managers. I cannot keep track of all these species. So we're really reliant on the help of others with this particular tool. And then finally, the INFRA-specific taxon protocol. Again, this is for cultivars, varieties, subspecies, hybrids. We want to make sure that the recommendations for the parent species are not the same for the cultivar. So we don't want to release something that's just as bad or worse than the parent species. And again, this is an internal tool that we're starting to expand this out to some other cultivars, but this one's written into the cultivar release process at University of Florida. And mainly I look at things like forage species and landscape gardening species like this Ruellia. You have a wild type over here, and then this is the improved cultivar, and here's wild type Lantana and Bloomify Red. So we want to know if they're going to be sterile. Are they going to behave differently? Are they going to revert? If they do produce seeds, are those seeds viable? Those are the kind of questions that we look at with that. We have over 900 species in our database. And if you go to the website, assessment.ifas.ufl.edu, you can filter that database based on what conclusions you're looking at, the zone that you're interested in, what growth form. So we do have that, all that data is in the database so that you can do filters to sort of tailor the list you want to look at. And then you can download those lists in Excel and manipulate that list however you need to use it. I want to go through the conclusions because we do have different language for depending on the tool. So we've color coded the conclusions to be like a stoplight. So red means don't use it, yellow means proceed with caution, and green means go. So for the status assessment and ITP conclusions, you're going to see this kind of language. Invasive, no uses, or just invasive. This means that it's not to be recommended by the university. So again, another important thing to note is that this is, the IFAS assessment is, our conclusions have to be adhered to by university faculty and staff. So they are not allowed to recommend invasive species or predicted to be invasive species. We aren't allowed to promote those species around the state as a part of our land grant mission. So invasive no uses means don't recommend this. Caution means you can recommend but manage to prevent escape. Not a problem species. You'll see documented versus undocumented. That's just whether or not it's present in a natural area. And this one is okay to recommend. For the predictive tool conclusions, you're going to see risk pop up on each of these. So high invasion risk, moderate risk, evaluate, and low invasion risk. And so the same implications for these, even though it's a prediction versus a diagnosis. So I'm going to shift and talk about a new project, or actually it's a year-long project now that we're getting ready to wrap up. So we got a grant from Florida Fish and Wildlife to conduct a horizon scan to try to identify species on the horizon. So most of the stuff we put through our program is are species that are on our radar in some way. So we took a framework from some researchers in the UK, modified that for Florida, and we're currently doing this horizon scan where we're using rapid risk assessment and consensus building to identify invasive species threats to our state. And this one is all taxa. So the context, current approaches employed, predict, regulate invasive species. Most of those happen after they've been introduced. And we really need to know and identify unknown or emerging invaders so we can try to prevent them. Remember that invasion curve, we want to catch these things before they get here. So this seems to be the most proactive approach. And Florida specifically is a great place to do this because we have 30 points of entry, airports, domestic and commercial ports that make us a gateway to the whole Southeast and in turn the United States. And then just a definition of horizon scanning, it's a method used to identify potential threats, emerging issues to inform policy, regulations, and decision-making. And we met in December of 2019. Here's a look at the fine group of folks we brought together out at Austin Cary. The workshop, we met for a couple days and really we did the presentations and used, our group got together and sort of tweaked that framework to make it fit our purposes. And we refined our methods. So we broke out sessions to each of the groups, and depending on if you were marine team, vertebrates, terrestrial invertebrates, aquatic invertebrates, plants, or vertebrates, somewhere between 80 to 200 species per group that we initially considered. We use a rapid screening tool that they take about an hour a species. So we just score 1 to 5, likelihood of arrival, establishment, and causing impacts, for a possible total score of 125. We also assigned confidence, or, it's like, I know that this species is gonna arrive in Florida in the next 10 years. I'm certain of it. So I put the highest certainty. But if it's likely, but I couldn't find a lot of evidence to support that, well, then maybe that would be a lower confidence for that. And then we also considered pathways. So if you're thinking about how a species gets to the state, we use this Harrower framework where you have dispersal. So you could have something that's just released in nature, it escapes in nature, or it just sort of, is a contaminant with something else that comes over in. So something gets inside a shipping container and gets released. Vector, this would be your transport stowaway. So think about things like sticking on the outside of a ship. And then movement of commodities. So this would be corridors, like the connectivity of corridors, or unaided. And unaided introductions are kind of rare, but just think about this coconut just floating from island to island. So here are our early contenders. This is, we've got the pathway at the bottom, but just for, here's 3 of the higher scoring terrestrial invertebrates, this species of cabbage moth, the nun moth, and cotton bullworm. For the aquatic invertebrates, we don't have zebra mussels here. Do we? Did someone peek in? Okay, I thought I heard something. No zebra mussels, no red swamp crayfish, but these both scored 125, the highest possible score. So these are a big concern for the aquatic invertebrate team. Marines, we have this colonial tunicate, bryozoa, and the Asian date mussel. These are some of your early contenders there. For vertebrates, we have this crab-eating macaque, Japanese fire-belly newt, and African clawed frog. In particular, this crab-eating, I get a lot of questions about this macaque, this monkey. And how could something like that get here? Well, this is a monkey that's used in animal testing and the medical testing. So the person who did this actual assessment was like, if this one gets out in Florida, we are in big trouble. So it is plausible that it could get here. This fire-belly newt, this is in the pet trade. So just something to be on the lookout for. And then here we are with the plants. Privet, colonial bentgrass, and Scotch broom were some of our higher scoring species. We ended up with, our group is almost finished. We ended up with 6 high-risk species that were then our next steps for this group in particular is I will take our high-risk species and our moderate risk species And then we're going to add those into the IFAS assessment, so we have that information available for folks in the public. So our next steps, we're finishing this that project up in probably the next, I think we set a date for mid-June to do the final consensus building. We're already writing some papers up and preparing reports for FWC. So hope to get those out there very soon. Again, our assessment website is assessment.ifas.ufl.edu. If you want to look up any of our 900 plus species, you can hit the contact me button. You can send questions directly to me. And I think I have, 3 or 4 minutes for questions now.
Yes, does anybody have any questions for Dr. Lieurance? If not, I will read the ones from the chat. The first comment from the chat is Regina said she uses the assessment site all the time and it's super helpful to help educate homeowners. And then she also had a question. She was wondering if a species has to be on more than 2 acres to be assessed.
That is specific to Florida, the Florida non-native planting rule. So essentially, if someone wants to plant, we've had some bamboo that people have been wanting to plant. There's been a history of some of the eucalyptus for biofuels. So if they want to plant more than 2 acres in production, they go to FDAX and fill out a permit. FDAX comes to me. That's the Florida Department of Ag and Consumer Services. They come to me and I do the weed risk assessment, and that's part of their consideration of whether or not they grant the permit, but there's other stuff that goes into that as well. There's surety bonds and site visits and things like that. So we're just a small part of that process, but it's really nice that the state is considering invasion risk when they're granting those permits. Anybody can request, if you have a species that you're seeing, there's a species request form on the assessment page. For the most part, it's things that are already in our assessment, and I can direct people to the results that exist. But if you have a new species that you're seeing, I appreciate the heads up.
And the next question is from Beth, and she says, for red, even though we can't recommend, are they still legally available?
So yeah, the red species, that's just, that is an internal, the IFAS assessment is non-regulatory. It's just for university faculty and staff, and then for education materials. We have the last few species that have been added to the noxious weed list, the state noxious weed list. They have incorporated weed risk assessments or status assessments that we've done. And actually the last 3 that have gone on the list, I petitioned. So, for something to go into that noxious weed category, that's a state regulation list that they do ask. They also, when they determine whether a species is going to be put on that list, they do bring in expert panel from the University of Florida, specifically Center for Aquatic and Invasive Plants. And usually I go in there as well if I'm not petitioning, I go in to answer questions. And there's an open public comment before things get put on the list.
Okay, did anybody else have any other questions? If not, thank you so much for your presentation.
Thank you.
And I think we'll go, Larry's going to talk about the DDIS and how, if you find something that you want some more answers about that could be invasive or a pest or a pathogen, how you can use that system to get a specialist to answer your questions.
Okay, good morning, everybody. I'm Larry Figart. Let me move over here so everybody can see there instead of a bright light. I'm Larry Figart. I'm the Urban Forestry Extension Agent here in Duval County. Just for a little bit for the Master Gardeners from Duval County, you might be interested. Fortunately, we are getting ready to interview later on this month for the environmental horticulture master gardener coordinator position here in Jacksonville. So that's good news. So let me go ahead and get started reporting things using DDIS. And so the first question might be, what is DDIS? It stands for Distance Diagnostic Identification Service. It's been around a while. We kind of think of distance things as being created by all this COVID stuff, but it's been a tool around for several years. And what you can do is, if you're identified as a user, and we'll talk about how to do that in a little bit, you can submit samples to where folks from all over the state can look at it and help you identify the sample. And so then if it's something that it's a pest, the experts will identify the problem and then even provide a recommendation. And so it provides data for research. So folks can gain priorities. They can, folks that might be specialists might not have any idea that this is in their part of the state. Also, extension agents might not have any idea that something is in their county. So this helps provide data for all sorts of things.
Takes blood from people. She, that's what she does. So she's constantly traveling and moving around.
So why is this important? Is because it provides a statewide network for us to be able to identify plant diseases, invasive species. It may even be just educational. You may have an idea, I get these things all the time. What kind of plant is this? And it may be something that's noxious, and it may be something that's completely harmless, but it just provides that portal, that avenue for folks to get things identified. And so what makes it important is it helps to make something an accurate identification. I'll give you an example. Somebody sent me an image the other day, and they were trying to eradicate it, and it ended up being a lady beetle larvae. And so they had no idea that this lady beetle larvae was something good, and they were trying to spray for it and kill it. So accurately identifying something is important in order to figure out whether it's something you need to control or not to control. And then kind of our reason for meeting this morning is that if it's something to where nobody else knows, or where maybe folks don't realize that it's in the state or that it's becoming a problem, it provides that early detection that is easier to control. It provides that early detection of invasive species, plants, animals, insects, so that it can be controlled while it's still small, while the population is still small. So what it's important, if you look at this kind of diagram here, the diagnostic network, the DDIS, it's kind of in the middle. And so all these folks, these little arrows that go outside, are folks that utilize the network. And then they are also connected to, like, the regulatory agencies. Florida Department of Agriculture is connected to plant inspectors and into farmers. County Extension, we're connected to you, and you're connected to your communities. So that's good as well. To expert labs, folks that can identify things, and then diagnostic clinics such as the Dr. Harmon's facility in Gainesville, and then folks that do external identification. It's connected to those as well. And the fact that it's web-based, mailing in hard plant samples or insect or disease samples is important, but this is also, because it's connected through the internet, it allows for something that may be a little bit quicker in identification. So what kind of things can you send in and send in images or photos through DDIS. And that can be a plant disease. It can be a sample of an insect, like we're talking about today. It can be a plant or a weed identification. One of the interesting things is that there's actually a fungus identification part to this, where somebody just wants to know what kind of mushroom this is. Just another side note, it was kind of important because somebody was wanting to know if a recent one that I saw was wanting to know if it was a poisonous mushroom or toxic mushroom. And so that was important there. Whether something is a abiotic problem, meaning it's something that's not related to an insect or disease at all, and it has to do with pesticides, fungicides, nutrient deficiency, that can be helpful. And then again, like what we're meeting here this morning, if it's something that we've never seen or we're tracking that might be a potential invasive species, or we know it's an invasive species and we find it in maybe a place or a county that we didn't know it existed. And then it's also important for farmers and livestock. Think about, now I don't know if we get into viruses, but think about the origin of this coronavirus that we're talking about now. It was, folks are talking about that it might have been in this market where animals were bought and sold. So DDIS is very critical in that. So how can, anybody can become a user of the DDIS. The ddis.ifas.ufl.edu is an important thing to bookmark. And so what you do is you connect to there and you click, there's a link that says become a user. And so when we get to that become a user, you'll have this opportunity to input some things. So just if we're master gardeners or homeowners or general public, when we get down here to where it says user group, right here, we'll put extension clientele, IFAS unit name. We won't put anything there because we're not, members of the general public are not part of an extension. For me, if it was me doing it, I would put Duval County extension. And then it gets sent to us and we approve the user or not. I don't know of any time where I've not approved a user. So then if you've got something you want to submit, you can go to your DDIS page, go to the DDIS page, and there's a section that says My DDIS. And in there, you can start the sample submission. Hopefully you've got an image or something you want to post. Hold on, let me catch up to my notes. I wrote some notes. I want to make sure. So at the top of the My DDIS page, there's My Samples, and that shows the images that you've submitted. If you want to add something to it, if you want to add some kind of edit to the form, this is the place you would do this. And then if you want to submit something that's brand new, this down here, submit a sample for diagnosis, is where you would start the process to submit a new sample. So when you get to the type, it may be a plant disease, an insect that's not associated with a plant, so to speak, a plant or a weed, mushroom, anything. But if you're not sure, then you can click as many of these categories as you see fit. And it's kind of better to, for sure, if you're not certain, don't worry about clicking too many categories. So then you can submit it in public or private. You can send physical samples. to the lab, and it'll give you an opportunity to write down to submit when you actually sent the sample. If you have images, sometimes those images are geo-located to where they actually have GPS coordinates on those, especially if you take it with your phone, it'll locate exactly where you took that sample. For instance, in Duval County, I found a huge field, and it's the biggest I've seen of kudzu in Duval County. And so I was able to send the images and then geolocate, exactly where it was on a map because the image said where I was. That's kind of scary, but it helpful in this instant. And then you can fill in as much information as you can on the sample, whether, what was the weather when you saw it? Where did you see it? Was it eating something? Was it on the blade? Was it on the stem? Was it on the root? Was it in the soil? And then lastly, you can click which extension agents. Hopefully, if you're a Duval and Nassau County extension agent, you pick me. But if you think there's another extension agent or other extension agents nearby that may be helpful, that's good. We have Chris Kerr, who's the commercial horticulture agent. He's got a doctorate in DPM, plant medicine, and he's very good at identifying things. So, and then any other additional information that you might think will help us figure out what's going on. And then you have the opportunity to add images, and this is just very easy. It can either you can either take it and click it and drag it to this spot, or if you hit click to add images, it takes you to your file system and you can find the image and add it. So once a sample is submitted and you send it to the extension agents, they can either, say exactly what it is, or, and this is what I do a lot, is we can send it on to the specialist. We can send it even further to the specialist, because sometimes, especially on some insects, I'm not the best expert, and as we saw from those moths that were listed as high priority, they look alike, and so it may be hard to identify them specifically. And then if your sample is identified, you actually receive notice that that sample was identified. So just a note about disease samples, many plant pathogens cause similar symptoms, and we're going to talk about that in a little bit, between a symptom and a sign. A sign might be is the actual physical thing that will help you that's connected right to that specific pathogen. But a symptom might be something like a witch's broom, or yellow leaves, or holes or spots on the leaves, things like that. And so there may be different diseases that cause similar symptoms. And so to be able to identify a pathogen from those symptoms, sometimes you need to go ahead and send in physical samples so that folks can look at it with a high quality microscope, or they can try and grow it out and see what happens, or even do PCR testing to look for the DNA of the pathogens. So that might be something to where you talk with your extension agent or specialist and try and figure out the best way and how to package up a sample to send it in. So just a little, these are a few helpful links. Your local extension office, hopefully folks in Duval County know how to get a hold of us. But if you're in a different county, or if you maybe in a state park somewhere and you find something in a different county and you want to alert them, you can find local offices this way. Usually typical, the best way I've found actually is to type in the county that I'm looking for and then type in IFAS behind it. And sure enough, usually the first thing that pops up is that county, that local county website. So for insect IDs, Dr. Lyle Buss has an excellent website to help identify insects. And then if we need to send something into the Plant Diagnostic Center, Dr. Carrie Harmon, typically there's a fee associated with a lot of these submissions. And then also there are certain ways to package them up. And so talk to your local extension agent and figure out how the best way to do that would be. The State Department of Agriculture has some submission forms. And as was alluded, there are state noxious weed lists. There's IFAS assessment. So there's different lists, but the noxious weed list is very important. Those are things that are illegal to possess, distribute, sell in the state of Florida. And then here's just some good contact numbers with the Department of Agriculture. DPI is Division of Plant Industry, and they primarily work with nurseries and producers and things like that. And this is just the citation for this program, and I can open it up if anybody has any questions.
There are a couple of questions in the chat if nobody has any that they wanted to say. The first one is from Beth, and she asks, when should a client use this system versus contacting a Master Gardener volunteer at their local extension office?
That's a good question. I think, you know, there's multiple people kind of doing the same thing. And I think Master Gardeners, if it's something that they're sure of, they can answer it. But if it's something to where there's any question as far as, you know, what it might be, what, you know, any, don't be so certain that you know it unless you really know it. And if there's any question, then pass it on to their extension agent or use DDIS to do it. You know, there are some things we see every day, and it's clockwork. We eat it, dream it, sleep it, but and especially for folks that are attending this session, there are things, you know, our state is with, I forget what it said, how many ports of entry we had. There's the opportunity for something to come in new every day. And so there may be some things we've never seen before. And so to recognize that and know that, that's a good thing for Master Gardeners.
And then piggybacking on that question is, Carol asks, is this a local process in our county extension office?
It's a statewide process, I think, is the way to answer that, but there's opportunities for local involvement, opportunities for local, you know, we'll get an email that pops up that says something has been entered into DDIS, and so there's a local opportunity to see it, to view it. I hope that answers the question.
Yeah, I think it does. And I think it has like a local aspect to it, but it also has a wider state aspect to it. And then, yes, Carol, we can provide the PDF of this PowerPoint so that you can click on those links. And then Also, Regina shared a link that will take you to a list that has all the diagnosticians on it. Were there any other questions about DDIS?
I just had one final comment, and this just has to do with, if you kind of think about how busy some of these folks in Gainesville are. And so I don't want to discount not sending stuff to them, but maybe first start out with your local county extension office, and then if they can't get it, then send it higher up the ladder. But if you can imagine how many millions of people live in this state and everybody starts sending samples to them, it can kind of get overwhelming. It gets overwhelming for me sometimes. So.
I've one more question. This is by and large a free service, except maybe for some of the advanced studies.
Yes, it's just if you have to send, there's certain rules, you know, as far as sending in samples to the labs. You know, some of the diagnostic tools are expensive, and so there may be some costs associated with that. But the DDIS is free.
Okay.
And just to add to that, anytime you really think that you have an exotic, we want to get that sample. And so we can make sure that sample is free if we think we have something really important. And Larry is just absolutely right. Our counties are the front lines. And besides, Larry wants to know what's going on in his county. And so it's really best to kind of go through the county first. Really an excellent presentation. Thank you so much, Larry.
And if there aren't any other questions, we can go ahead and split back up into our breakout groups, and we'll be doing the Build-A-Bug activity. Okay, it's 10:30, so we can go ahead and get started again. Brad is going to be talking about the Asian longhorned beetle, and then following that, we'll be having another activity.
Excellent. Well, good morning, everyone, and thank you, Shannon. Again, my name is Brad Danner, and I'm the State Survey Coordinator of the Cooperative Agricultural Pest Survey. And today I'm going to talk about a pest that my program is looking for in Florida, the Asian longhorn beetle. And especially for you folks in Duval County, this one is getting kind of close to home. I'll talk about that in a little bit. But this is a forest pest, again, called the Asian longhorn beetle. So in your breakout session, your group leaders probably discussed with you some of the features that beetles have. And we will be going over some of those features more specifically pertaining to this pest and how we identify it. This beetle is native to China and Korea, and was introduced in the US, probably most likely through untreated wood packing material. So wooden pallets or boxes that have gone through or have bypassed or possibly missed the fumigation treatments and things like that those materials are supposed to go through before coming into the United States. And so we've had a few incidences in the United States pop up, and I'll illustrate those in just a little bit. But this beetle can be a problem for both urban and suburban areas and natural areas such as state parks. You know, this can impact the lumber industry, hardwood lumber industry. For those of you who love your maple syrup, this one can be a problem for that as well, because one of its preferred hosts is maples. It also goes by a couple of other names. Asian longhorn beetle is the most common, but starry sky beetle is another term that is used for it. Anyway, this is just a look at the distribution that we've seen in the United States. You can see in Illinois, is specifically in the Chicago area. It was introduced and then later eradicated. So the Chicago or the USDA combined with some agricultural entities in the Chicago area really did a good job at eradicating this thing from Illinois. It's also been detected and regulated in Ohio and New York. And then what I mentioned earlier, our most recent detection of ALB, that's what we call Asian longhorn beetle for short, was just this past summer, 2020 in Charleston, South Carolina. That particular situation right now, I don't know if there's any quarantines in place at the moment, but my latest information tells me that there's about 4,000 infested trees in a 59-mile radius surrounding the Charleston area. And so this one, as you can see, as the beetle flies, so to speak, or as the crow flies, is not that far from where you all are located in Florida. And so my team actually, we've been surveying the Jacksonville, Duval, and Nassau County areas quite heavily ever since we found out about the situation going on in South Carolina. They are currently dealing with that. I believe they're trying to eradicate, but 4,000 infested trees in 59-mile area is a pretty large undertaking. So we're hoping that they can take care of that. Some host plants for the Asian longhorn beetle. Preferred hosts, as I mentioned before, maple is kind of the primary preferred host. So your sugar maples, your red maples. So it can have an impact on the maple industry up north. Elms, buckeyes, willow, and poplar are also preferred hosts. Potential other hosts, we have birch and sycamore. And then occasional hosts, we have green ash, mountain ash, mimosa, and katsura. Another thing that we've learned from the South Carolina situation is that they are seeing Asian longhorn beetle on Tupelo. And so we do have some dense Tupelo populations here in Florida, especially in the Apalachicola River corridor of the Panhandle. So we're going to be keeping an eye on that area as well. Some external signs of Asian longhorn beetle. These are kind of the things that, the beetles themselves are, I'll talk about their size in just a minute, but they're larger. So they are visible to the naked eye, very much so. But a lot of times before we see the beetle, we'll see damage associated with an infestation. And so some of the external signs of Asian longhorn beetle are the oviposition pits. These are areas that can be seen on the bark where the adult has kind of scratched or chewed away, and that's where they lay their egg. So they'll scratch a spot, they'll lay an egg, they'll go to another spot, scratch another pit, lay another egg, and so on. Frass or shavings. Frass is a mixture of the beetle's excrement combined with maybe sawdust or other particles associated with the host plant, or shavings from the chewing and scratching of the wood. If you were to peel away the bark, or if there's enough damage that the bark falls off naturally, you would potentially see these feeding galleries underneath the bark, where the larval stage of the beetle has tunneled through the wood. And then external, or the exit holes. These are, as you can see next to a dime here, they're pretty good size, probably about 1/4 of an inch. You can stick a pencil into these pretty deep. And this is the adult exit hole. After the After the pupa has emerged as an adult, the adult will exit the tree and start the cycle all over again. And so a lot of times you'll see exit holes like this. So the larvae, they feed in these galleries and they feed on the wood under the bark, but the adults feed over here, the adult feeding damage. They will feed on the leaves, the petioles, or even some of the younger bark tissue of the branches. And so you can kind of see this kind of scratched away material looking area on the bark where the adults have fed on the young wood. Internal feeding signs, again, galleries and tunnels in the wood. A lot of times to see this, you have to split the branch open or peel away the bark to see what's going on there. And then pupal chambers. As the larvae is reaching the next stage, they will kind of carve out this pupal chamber and seal it up with frass and shavings to kind of seal themselves in and protect themselves. And then they go into this pupal stage, which, kind of freakily kind of looks like an adult encased as a mummy. And they go through their metamorphosis there, and then they emerge as an adult beetle. Symptoms of an Asian longhorn beetle infestation. This is going to be the tree's kind of response to the attack from the beetle. So sap flowing from wounds in trees. I mean, you may see this with a lot of other pests, not just Asian longhorn beetle that does this. Any insect that bores into a tree can potentially cause one or more of these symptoms. But sap flowing from wounds, the oviposition stains, so those oviposition sites that I mentioned earlier, as the wood oxidizes, it will change color to like a bronze or a brown or even black as that spot ages. Cracks in the bark, you can see this bottom picture, the arrow is pointing to some cracking in the bark associated with an exit hole there. So, the damage that these the larvae and the adults cause can cause some cracking of the bark. Callus tissue around wounds. A tree's response to damage is to almost create like a scar like humans would, except it's callus tissue. And sometimes that callus tissue can hide some of these other features that we look for. More obvious or signs that you may see more regularly, yellowing and drooping of leaves or branch dieback and tree death. And for a tree to die after an Asian longhorn beetle infestation can take, 5 to 10 years as the population builds up inside the tree. But the key here is it's going to be top-down death. So as you can see on this picture on the right, the branches at the top are dead and the branches at the bottom still seem to be living, at least for now. And why this occurs is as the larvae are feeding in the phloem tissue, it's disrupting the nutrient flow up and down the tree. And so nutrients that are being taken up from the soil can't reach the top because those pathways for travel have been interrupted by the feeding damage. And so you get this kind of top-down dieback of a tree. So the life cycle of Asian longhorn beetle, at the top here, we have the egg. The typical life cycle can take 1 year all the way up to possibly 2 to 3 years in colder climates. But again, we get these oviposition sites where an adult beetle will lay an egg. That egg will hatch in roughly 10 to 15 days, at which time the young larvae will begin tunneling into the tree and start causing the damage by feeding on the inner bark and the outer sapwood. And this occurs for about 20 days or so. And as the larvae mature, they will tunnel further into the tree and feed on the tree's heartwood. And then this is really where a lot of the damage to the tree can occur. It can affect the structural integrity of a tree. And this feeding in the heartwood can occur for several weeks to several months through the fall and winter months. So you think about larvae inside a tree, they're protected from the elements so they can continue to feed. When they are ready to pupate, they will crawl closer to the surface and they will create that pupal chamber that I illustrated earlier. We have the pupa on the bottom here. Again, it kind of looks like an adult already encased in like a mummy type of situation. They will remain in that pupal stage. Sometimes they'll overwinter that way, but it can take as early as 20 days for them to transform into adults. And then the adult here on the very left will emerge and they will do some feeding, and it takes them, about 2 weeks or so to reach maturity, at which time they can mate and lay eggs and start all over again. So identification of the Asian longhorn beetle, again, as far as insects go, they're pretty large. The eggs themselves are 5 to 7 millimeters long. So for an insect, that's pretty large for an egg. They're usually white when they're laid and turn kind of a cream color or off-white as they mature. And again, those can be found in those oviposition sites on the bark. The larvae themselves are cream colored and they have a head capsule. It's kind of like a little protective head capsule, but that contains the mouth parts or the mandibles, the chewing parts that you learned about earlier. And so they create their damage by chewing through the wood. The young larva itself can range anywhere from 7 to 20 millimeters, and as they mature can reach 30 to 60 millimeters in length. So they get rather large. You find these kind of big grubs, so to speak. The pupa themselves are an off-white to ivory white. They're about 30 to 37 millimeters long. And if you were to break open the wood, those could potentially be found in that pupal chamber. The adults, you can see on the left and right here. On the left, we have an adult male. And on the right, we have an adult female. They're kind of this glossy jet black color with the white spots or speckling. Just there's no real pattern to it. It's just kind of random how the speckling shows up on their body. And they have these really, really long antennae. Kind of one of the differentiating features between the male and female is the length of the antenna. The adult female, the antenna is about the length of the body. And in the adult male, it's about 1.5 times the length of the body. So typically the males will have much longer antennae. But the adult size, males range from 22 to 36 millimeters in length, and females are, and females are just a little bit smaller than that. So now getting to the important parts, the lookalikes. What differentiates the pests that we don't want versus the pests that we might already have or natives? So we're going to cover a few here. And then in the next breakout session, we'll kind of test what you've learned about these lookalikes. So on this slide, we have two, the southern pine sawyer and the pine sawyer beetle, both in the same genus Monocamus. So they're in a different genus altogether than Asian longhorn beetle. But the southern pine sawyer attacks dead and dying or down and freshly felled trees. So the Asian longhorn beetle is going to attack living trees. The southern pine sawyer is going to take advantage of situations where a tree is possibly already dead. Another big difference here is these two lookalikes attack pines, whereas the Asian longhorn beetle attacks hardwoods. So you're not even going to find these pests in the same host material. The southern pine sawyer kind of has a mottled gray or brown body with a strong spine, illustrated here by the yellow arrow. And they're going to have very, very long antennae. So long, in fact, that in this picture, they're kind of broken off. You can't even see the entire antenna. The pine sawyer beetle, all the way on the right here, is going to have a brownish body with irregularly notched paddles of brown on the elytra. So remember in your build-a-bug activity that elytra are the hard outer wing coverings that occur in beetles. And that's where we find some of these diagnostic features. And then again, they're going to have extremely long antennae by comparison to the Asian longhorn beetle. A couple more lookalikes that we have here. The white spotted sawyer, which is also in the genus Monocamus, and the cottonwood borer. Starting with the white spotted sawyer, that one is going to also use conifers. So again, we're looking at a different set of hosts. They will lay their eggs in dead or dying conifers. Their body's going to be slightly smaller than ALB, ranging from 13 to 22 millimeters. And they're kind of have a kind of bronzy black overall coloration with a dimpled surface. So I don't know if you can see that on the slide, but the yellow arrow is kind of pointing at this dimpled surface on the elytra. And then they're going to have a large white spot between the base or the top of the base of the elytra, where the red arrow is pointing there. And then females also have a distinct white patches that cover the wings. So you can kind of see that in this picture. And then all the way on the right, we have the cottonwood borer. The way I kind of think of this one here is it's got a white base with black spots, whereas the Asian longhorn beetle has a black base with white spots. So they're kind of opposites. And then the cottonwood borer, that pattern is going to be kind of more regular than Asian longhorn beetle. But their antennae are extremely long as well, much longer than the body. They have black rectangular areas over a yellow or creamy white body. And they're commonly found in cottonwood, willow, and poplar trees. The thing to keep in mind with these two here is that the white spotted sawyer is very rare, and the cottonwood borer is not going to be very common in Florida. Another lookalike we have here is the long-jawed beetle. These guys are also not native to Florida. However, they have some limited distribution here. Trichoderes mandibularis gets its name because of its extremely pronounced mandibles. As you can see in the picture, they have very, very large chewing mouth parts. They are native to the western part of North America, in California and southern Texas, through the Yucatan Peninsula and Baja California. But a population was found in the lower Keys several years ago. And also in 2004, it was found in Manatee County, kind of over in the Tampa Palmetto area. They breed in dead or dying branches of hardwood trees, including citrus and willow. And a lot of the specimens that were collected in Florida have actually been found on traveler's palms. And then the last lookalike we have here is probably the closest looking to Asian longhorn beetle. In fact, it's in the same genus, the citrus longhorn beetle, or CLB as we call it. This is also a pest that my program is looking for. This particular one, really the biggest difference between the two is indicated here by the yellow arrow. The citrus longhorn beetle has these elongated tubercles or short processes at the base of the elytra called granulae. That's the biggest difference between the two of them. The host range for these two is very, very similar. Essentially, citrus longhorn beetle has the same host range with ALB, except for you can also add citrus to that list. In this particular case, if you see this one needs to be reported also. This is one of those beetles that's on the national priority list right alongside ALB. So if you see anything that looks like this, you would want to report it. Monitoring for Asian longhorn beetle. Really, there are no suitable traps at this time. The USDA has been researching some trap and pheromone lures to use, but so far nothing has panned out. So our best way to monitor for these things is visually. You know, you can do surveys from the ground of the canopy, looking for that top-down dieback or droopy leaves that I mentioned earlier. If you are so inclined and have the resources, a bucket truck would be beneficial. That's generally kind of what we pitch to utility companies and things like that, you know, to keep an eye out when they're up in the canopy. Even smoke jumpers here on the left. I don't know anybody personally that surveys for Asian longhorn beetle this way, but it would be a way to get up into the canopy and look at things. So we're visually looking for the signs or symptoms of ALB or the adult beetle to be present. Management, basically, one of the big things that we push is not to move the invasive species around. That's one of the best ways they can move. I mentioned the wood packing material earlier on. Locally here at home, we can, you know, not move firewood from one state to another, especially snowbirds. We encourage them to leave their firewood at home and just buy their wood where they're going to burn it. So try to leave your pests where they are and not bring them to Florida. Prevention, when we do find any of the pests that we look for, but especially Asian longhorn beetle quarantine infected areas. New York, Massachusetts, and Ohio have all had quarantine areas. I suspect, even though I haven't confirmed, that South Carolina probably has a quarantine in place around their population. But I have not confirmed that. But if they're trying to eradicate, I imagine they have a quarantine area. Other control measures in infested quarantine areas, you know, certain chemicals can be used, but this is expensive, so it's not used as a preventative. It's only used when there's an active quarantine situation going on. If a tree is infested, it must be removed and destroyed. And it is not recommended to move infested material to another location. It should be handled on site, and the best way to do that is with a chipper. And treat wooden packing material from infested areas. If you have to move product out of an infested area, it needs to be treated before it's moved. What should you do if you think you found ALB in Florida? Take pictures, collect the specimen. Please don't destroy it. We've had people report ALB before that we've had to follow up with, but they don't have a specimen because they've smashed it and thrown it away. The only way we can really confirm something is if we have good pictures or a specimen to support the report. And get the samples to your experts. You know, if you need to report it in Florida, your IFAS faculty should be your starting point. You saw these links earlier. I'm just putting them up here again for reinforcement. Find your local county extension office, work with them, get your specimen to either Lyle or Dr. Carrie Harmon. And if it is ALB, they will report it to FDAX DPI, which is where I work, and we'll get the appropriate regulatory actions going. I'm not going to go through the DDIS very much. That was covered very well earlier, but this would be a way to potentially report ALB if you think you have it. The FDAX, the Division of Plant Industry, it's the Florida Department of Agriculture and Consumer Services, Division of Plant Industry. We are the regulatory agency in Florida for this kind of thing. It is our responsibility to announce the detection or establishment of any new invasive species. So we would have to confirm the diagnosis of ALB in order to make that announcement. And we do have a legal obligation under Florida statute to make that known. Here again are the links. We have a specimen submission form that we use to make a diagnosis. And then the link at the bottom here will take you to our FDAX page where you'll find a series of videos and documents that show you how to submit a sample for identification. These are just reinforcing the contacts. Again, Dr. Leroy Wilby is our bureau chief of our diagnostic bureau here at DPI. And the assistant chief is Dr. Paul Skelly. Actually, until about March or April, Dr. Paul Skelly is the contact because Dr. Leroy Wilby is deployed with the military in Afghanistan and will return in about March or April. Then also, if you can't get a hold of either of them, the Division of Plant Industries hotline is here. You call that number and they will route you to the appropriate person you need to speak with concerning the issue that you have. And then this is just some information on this presentation. It's been continually updated as we learn more information about ALB. The most recent update coming in November of 2020. And some of the original authors, and I can open it up for questions.
That was really great, Brad. We really appreciated it. Actually, we're going to need to go directly into the breakout, but there will be sometime at the end of the overall session for questions. So I'm going to encourage everyone to continue to talk about the ALB during the breakout. And for the breakout leaders, you'll have 10 minutes for your breakout. Thank you so much.
All right. Hello, everybody again. Hope you had a good, quick activity with the ALB and could spot some of the differences between the lookalike species. Real quick, before we go into our last presentation, I wanted to talk about the goodie bags that were advertised in the flyer for this workshop. And can you guys see my screen? Okay. And so I'll be emailing you after this workshop later today or tomorrow with more information. And I put in the chat bar the surveys, one's an attendance survey, and that is going to be how we can get your information to contact you about getting the goodie bags. And then also the other one is the exit survey. And that's the information for us so that we can make the workshop better for you guys in the future. And in the tote bag, you're going to get an invasive pest ID guide and then so it'll be a little quick book that you can take with you. And you also could contact Larry about getting coupons to send samples to the Insect ID Lab or the Plant Diagnostic Center. So we'll have those free to you, which normally it's a $40 value to submit something to the Plant Diagnostic Center. So we have those coupons available. And then you also will have a field sampling kit. So it's in a handy dandy fanny pack to go with you wherever you may go. And you'll get Hans lens and a couple of sample vials and Ziploc bags so that you're able to sample for some pests and then take a closer look at them. And then we'll also be sending a tube so that if you need to ship your sample to Lyle at the Insect ID Lab, you'll be able to do that. And speaking of submitting samples, the first 20 people to submit a sample will receive either a phone microscope attachment or a hand loop. And then also somebody who participates in the exit survey, so somebody will receive a hat at random. So there's a lucky winner somewhere. And just again, I put it in the chat. I'll add it. I'll keep uploading it so that it'll be at the bottom of the chat. But there's an attendance survey, so we know to get you a goodie bag. And then there's the exit survey, so we know what we can do to improve in the future. And they both, I don't even think they take 10 minutes. It might take 10 minutes for both. So we just really appreciate you letting us know how we did. And I think with that, we can go into Dr. Hodges' presentation and last talk of the day.
So thank you, Shannon, for that. And it's great to be here with you. Glad to be here with the last presentation for the day. And we're going to talk about some new invasives that have just arrived in Florida and some ways that we can be on the lookout for invasive pests as we conclude. So it's really important when you think about your role in protecting Florida from invasive plant pests, you really want to make sure that when you travel, you don't pack that pest. Brad Denner talked a little bit about this with the firewood example. Firewood is like a #1 example of how insects, in particular, different types of wood beetles, have sometimes moved to new areas and caused quite a bit of devastation. And so it's important that you not bring those pests with you. So I assume some of you have probably traveled not in the last year, but maybe prior to that, and you've seen these "don't pack a pest" signs. They're in the airport. There are videos that can be seen at different places. And it's really important not to bring fruits and vegetables, plants and cut flowers, meat and animal products, and live animals from other countries without a permit, because those organisms can actually cause disease in the plants and animals that we actually have here in Florida and throughout the US. So it's really important that you're not part of the problem in terms of actually bringing or smuggling some of these materials into the US that are prohibited. And I think oftentimes people bring these materials into the US not realizing it could be a problem. But for example, if you're traveling from somewhere that has Mediterranean fruit fly and you decide to take an orange from that country and somehow it makes it through the airport with you, you get it home and you cut into this orange, you notice that, oh, well, I don't want to eat this. It has maggots in it. Well, those maggots could potentially be an exotic pest that we don't want to have in the state of Florida. So it seems harmless to have a piece of fruit, but in fact, it can be quite problematic to bring fruit or meat products from other countries to the US, because there are these foreign diseases, foreign animal diseases, as well as invasive plant pests that we don't want to introduce into our environment. You've probably also seen the detector dogs at some of the airports when you used to be able to travel, and hopefully we'll all be traveling again soon. But these dogs are actually trained to sniff out agricultural products. So they can tell if you have fruit in your bag, and they can even sometimes detect snails and specific organisms that they're actually trained to sniff out. And so really don't bring the pest with you is key. And when you think about Florida and why so many species are introduced, it's really because of our favorable climate. We also have more airports than any other, than many other states, maybe not than all other states, but we certainly have 12 major international airports. Florida receives more cut flower imports in Miami through air cargo than any other airport in the United States. And this is information from U.S. Customs. So this is pretty significant. We also receive, we have more pest interceptions that occur related to cut flowers. And since we receive more cut flowers than any other location in the United States, it's not really a surprise that we have more pest interceptions. We also have deep water ports throughout the state of Florida. And this is another way that products are brought into Florida, but also with this movement of cargo and products and people, there's a good chance of things moving into Florida. Now this one with your location in Duval County, you're probably very familiar with these agricultural interdiction stations because you're very close to the Florida-Georgia border. And so these agricultural interdiction stations are managed by the Florida Department of Agriculture and Consumer Services Division of Plant Industry. And their goal is really to stop pests that might be moving in from other states to Florida, from like Georgia, or maybe even shipments of products all the way from California over to Florida. And so these are really important in terms of stopping pests there as well. Specifically, we've had numerous interceptions of the brown marmorated stink bug at these interdiction stations, as well as the Figart bug at these interdiction stations. And these are both two types of stink bugs. We do not have the Figart bug established in the state of Florida, but we do have a small localized established population of the brown marmorated stink bug. And we'll be talking a little bit more about that in a moment. So with the live plant material that we have through airports, deep water ports, and interstate travel, and our tourism industry, which is fairly significant as well as our long growing season and favorable climate, Florida is ideal for many of these invasives. So when you look at this, and maybe you could use the reaction button or something like that if you're familiar with it, or comment in the chat. When you look at these photos, what do you think you see? Would you be concerned? You could just maybe type in the chat yes or no, whether or not you would be concerned. And so basically what we're looking at, get another photo here. So the photo on the right is actually sea grape. And the photo on the left, I think, is some sort of beach bean. And so this is on the beach in Miami-Dade in South Florida. And you look a little bit closer, this is the insect as well. Now, is this the same thing as the photo on the right? Again, you can type in the chat, or you can, that would probably be the easiest thing. So those are actually two different things. If you look a little bit closer, this is a different type of insect in its adult stage. So this on the left is a type of plataspid. And some of you may have heard of the bean plataspid, which we have on kudzu and bean throughout Florida. And it was first introduced in Georgia in the late 2000, around 2009, but it also occurs in Florida. So this is a relative of the bean plataspid, but this is actually a true stink bug, which has, you can see if you were able to see the picture a little closer, you would see the scutellum. So when we were looking at this particular insect was first noticed by an observer on iNaturalist. And so I wanted to bring this to your attention, because you never know when you're actually going to find a new species to the state of Florida, something that's a new invasive, just in your daily walk and looking at photographs and things, places where you would normally walk and be. And one of our graduate students actually noticed that this photograph was on iNaturalist, and this is actually the black bean bug, Brachyplatus sylvaneus, and this was a new record for the U.S., and it was fairly significant from that standpoint. We still don't know how wide this particular invasive species has spread, and there haven't been that many reports of this particular species since it was detected in the fall of 2020. So I wanted you to just wanted to remind you to keep on the lookout for things that may be unusual. And so in this case, a teacher was just walking along the beach, took a photograph, saw something unusual on sea grape, and it ends up being a new record for the state of Florida. And so that certainly could happen with you as well when you're looking at your plant material. And it's important for us to know when these new species do occur in Florida. And what's interesting about this, now, when you look at this, maybe you could comment, do you think that this is an appropriate article, or do you think they're both pretty good in terms of the information? So if you look closely, there's just a little bit of text on this Miami article that talks about the teachers seeing the black bean bug on the beach in Miami. And then if you look at the article on the right, you see this new species of invasive crop destroying bug found in Florida. And then it has a picture of black bean aphids. Now, all of you are expert master gardeners, so you already know that a stink bug relative is not an aphid. So again, be careful where you get your information and make sure the information is accurate. That's also why we need to think about submitting our samples officially for identification, because when we do have these new reports in the state of Florida, we do need those reports to be officially confirmed. Thrips. Thrips are a particularly difficult group of insects. They're only a couple millimeters in size. And almost everyone, even trained entomologists, actually need to submit samples of thrips for identifications when they occur. I wanted to bring your attention to a few resources. This is a free online deck where you can look at some of the common thrips in terms of their photographs and damage that they cause to plants. We also have some really good extension information on thrips that can help you think about scouting for thrips and what the general life cycle would be for thrips, as well as management publications and pest alert publications. But the Asian bean thrips is another new invasive species, again, more specifically to bean. But in areas where it's native, it's actually native to Asia, it can cause 30% crop loss. And so when these new thrips come into Florida, things like this do need to be identified, confirmed. If possible, we would like to eradicate many of these pests when they first arrive. In the case of the Asian bean thrips, it has already spread too far to result in eradication. It's currently causing quite a bit of problems for our specialty bean growers and snap beans as well in particular. So it's just something else to keep in mind. And I wanted to remind you of the pest alert site on the Florida Department of Agriculture and Consumer Services website that is available for a lot of information about many of these pests. So now we're going to talk about one that's maybe a little bit more familiar to you. Some of you have probably heard of the brown marmorated stink bug if you're from more northern areas, but it was first detected in Pennsylvania in 2001, although it's thought that it actually occurred in Pennsylvania in the 1990s and was only identified in 2001. It causes this piercing, sucking damage that we see on fruit, and new foliage growth can also see some chlorosis damage. But it's become a major economic pest in many areas. Just last year, it was more of an economic pest for Georgia, and so it's had a slow movement down from Pennsylvania to the south, to the far south for damage. So it's a lot. It's very damaging because it's highly prolificous, which means that it has several hosts that are susceptible. What's really interesting and important about this particular pest is that we have detected an isolated population of the brown marmorated stink bug that seems to be reproducing in Lake County. We also think we found it established in Leon County. So on that slide where I mentioned all of those interceptions that were happening at the interdiction stations, those interceptions, with enough interceptions that happen at these interdiction stations, eventually the pressure of the pest is such that in many cases it can establish, as it seems to be the case with brown marmorated stink bug. The brown marmorated stink bug is also a hitchhiker bug, which means that it clutches on to like RVs and vehicles, or in the winter in particular in the northern states, and then it is able to find its way to Florida. So we don't think it's established in Duval, but if you do think that you have it, you can certainly report it and let us know. There's just some general information about the life cycle. We're going to just move on. Again, the brown marmorated stink bug can be easily confused with some of the other stink bugs that we already have in the state of Florida. Some, such as the spine soldier bug, are considered beneficial. And lastly, I just wanted to mention, as it relates to firewood, I wanted to make sure that you are aware of some of the information we have on the Protect US website. And this is an example of the red bay ambrosia beetle and laurel wilt disease, which has been particularly devastating for our forest. And actually, this beetle that causes this disease, and the disease is a fungus, has decimated our forest, really destroying much of the red bay that's in the understory. And this is the pathogen. And it's just a really small beetle that's capable of doing this. And it had moved around the southeast largely through firewood. And we just don't have as much of the hosts left as we once did, simply because of this disease. So really what you can do to stop the spread of invasives really makes a big difference. And with that, I'd like to thank everyone for your attention, and thank you, Shannon.
Thank you. And that concludes our Florida First Detector workshop. Did anybody have any questions for Dr. Hodges? And I re-put the attendance and exit surveys links in there. So there's a separate link for attendance and separate one for exit survey. And if you can get both of those in, that would be awesome. We really appreciate the feedback from you guys. And I know you all want your nice goodie bags, so that would really help us out.
And I know that there was so much information, but we really appreciate your time today. And anytime you have some questions, we would be happy to help. It looks like we have a question. Is there a good website to see recent invasive species? Probably the Pest Alert blog. We can send you information about that, as well as the FDAX Pest Alert would be a good source for information. And we have much of the content that we presented here is available on either the Florida First Detector or the Protect US website, which we can send you those links as well.
Thank you, Amanda. Looking forward to seeing them.
Yeah, that's great. We really appreciate your time and attention. And who knows, you might be the next person, just like the teacher on iNaturalist, that finds something new in the state of Florida. We hope not, but if you do, we hope that you will report it, and we can get that identified and know what it's doing in the state. Larry, did you have anything to say in conclusion?
No, other than, and I just want to confirm with Shannon, if we're going to do it similar to what we've done before, and that the goodie bags will come here to Duval County and then I'll disseminate them. Is that accurate?
Yes, that's the plan. I know there's a couple people from other counties, so that's why I need to get the attendance survey, so I know how many to send to you, if I need to ship some separately. But yeah, that's the plan.
Okay, thank you. Thank you, everybody.
We really appreciate your time, and the speaker is Dr. Lieurance, Mr. Brad Danner, of course, all of our group leaders, Larry, for hosting us, and all of you, the wonderful master gardeners of Duval, for your time and attention today. Thank you so much.